Sanger sequencing takes advantage of DNA's replication mechanism. An enzyme called DNA polymerase is involved in replicating the DNA, as discussed earlier. DNA polymerase works by aligning complementary base pairs, like T and A and T, and C and G. And so the replication progresses forwards. Scientists have been able to exploit this particular process with specialized nucleotides, such as this one here. At this particular A, the replication is terminated. The same at this particular A and this particular A. And so, with this in mind, scientists go through rounds of replication while exploiting this. Scientists can provide all of the conditions necessary for replication to occur, except they alter one of the sets of the nucleotides, in this case A. And so, in each position where there is an A, the replication is terminated, and so it can be inferred which positions contained a T complementary to the A in the original sequence. Let's say that was round 1, and so in the next round, a different specialized nucleotide will be used to infer the positions of its complementary base in the original sequence. Round 2, the same can be done for Cs to infer which positions had a G in the original sequence. Round 3, and for G to infer where the Cs were in the original sequence. And round 4 for T to infer where there were A's in the original sequence. Sequencing technology has come a long way since Sanger sequencing and continues to improve. Sequencing techniques have become faster, and the data systems have become capable of handling greater amounts of information. Thanks to the advances in technology, in 2003, the entire human genome had been officially sequenced. All of the chapters had been read. To say that we have read the entire human genome is not entirely true, as there are some areas that are still in question due to their repetitiveness. Let's say a region of the genome is constituted by a CA that's repeated thousands of times. It's difficult to tell the length of such regions, no matter how advanced the sequencing technology is. Such repeated regions vary in length between individuals in a species. Any two individuals of the human species are only 99% genetically similar. Hence, there will be 1% variation between any two individuals' genomes. A portion of that 1% can be accounted for by the difference in length of such repetitive regions. That aside, it is reasonable to say that the human genome is for the most part known and interpretable. And so this leads to the question, now that the human genome has been sequenced, what to do with it? Let's start by establishing that just because we know a sequence doesn't mean that we know what its function is. Thus, since 2003, scientists have been mining through the now known human genome in search of functional regions and possible causes of disease, finding genetic links to diseases that before then may have been believed to be completely independent of an individual's genome, such as obesity. GWAS, Genome-Wide Association Study, is the name given to the project, whereby scientists search for mutations that show association with different diseases. This is an example of what such research can produce. We see the y-axis measures the degree of association with whatever particular disease, in this case, coronary artery disease, which happens to be one of the main component characteristics of obesity. And on the x-axis, we see the chromosome numbers. And so the function of this plot is to depict the degree of association that mutations that span across the entire genome show with coronary artery disease. Hence, observe that a mutation on the 9th chromosome shows the highest degree of association. And yet notice that it is not the only one. There is a smattering of mutations across the entire human genome that show association with coronary artery disease. This suggests that the genes that control this trait are dispersed throughout the genome. One could speculate that the genes that control blood lipid levels, clotting factors, the construction of blood vessels, and many other things, all of which, if perturbed by a mutation, could lead to coronary artery disease, are unevenly distributed throughout the genome. This is just one example of how genes can be chopped and changed and dispersed throughout the genome through evolution. Sometimes genes are changed beyond recognition in comparison to the genes that controlled similar traits in the human genome's ancestry.